The neuroglia are kind of the unsung heroes of the nervous system. They're not actually neurons. Instead, they're glia cells, and they provide support to the neurons, but they're also very important in some of the signaling function that occurs, especially in the central nervous system. Due to the nature of this class and the fact that we don't have time to talk about everything, I'm only going to superficially cover the neuroglia. But if it's something that is interesting to you, I suggest that you talk to me and we can actually explore these in much greater detail. They're very fascinating cells. Four main types of neuroglia that we're going to talk about include the ependymal cells, the microglia, the astrocytes, and the oligodendrocytes. These are in the central nervous system. Now, I'm going to also talk about Schwann cells, and satellite cells, these are part of the peripheral nervous system, and we'll talk about those in a later slide. Ependymal cells are interesting cells that line the ventricles of the brain and the central canal of the spinal cord. Their primary function is to assist in the secretion of the cerebral spinal fluid. But they're also interesting and important in that they hold a very exciting promise for neuroscientists. Most of you should already know that once a neuron dies, it's gone. We don't really regenerate neurons. Neurons' death is a normal occurrence as we age. I have fewer neurons today than I did when I was 25. And we don't in general replace them. A brain injury or spinal cord injury is all the more serious because of our inability to replace those neurons. But recent science, and now in science recent can be 20 years, the way science works, the way science moves, you get this exciting discovery, and then somebody has to replicate that, and another person replicates it, and somebody else contradicts it, and then it goes on until eventually we end up building a consensus. But the exciting science is that there are actually these stem cells found in amongst these ependymal cells that may very well be capable of regenerating neurons. We don't really know how to unlock these yet. We don't really know how they get activated, how it works, but we do know that they're there, and it offers promise in stem cell research and that we might perhaps be able to harvest these cells, and if we can figure out how to make them work, we could replace neurons for individuals such as somebody suffering from Parkinson's disease where the neurons are dying, Alzheimer's disease, spinal cord injury, brain injury, all potential things that could be treated with these stem cells. Now the microglia are interesting. These are these little guys that crawl around. Let me see if I can find it in this picture. Here's one right here. And they are related structurally to macrophages, which are found in the immune system. They're not quite macrophages. They have other types of function, but their job is to be the immune system for the nervous system. You'll recall from your anatomy that the brain and spinal cord are separated from the rest of the body by what we call a blood-brain barrier. And this is designed to protect the nervous system. It isolates the nervous system from the rest of our bloodstream and provides this extra layer of protection for the brain. As a consequence, most of our immune system does not interact inside the central nervous system. We don't really get B cells or T cells or neutrophils or any of those cells there. In fact, most of the work is done by these microglia. Now, the exception to that is in a traumatic brain injury where this blood-brain barrier has ruptured. If you are doing a scan of a brain, if you do a tissue sample or anything like that, and you notice that you have other types of immune cells there, then you can be certain that the blood-brain barrier has been damaged at some point, and the immune cells were able to enter into the brain and spinal cord tissue. Astrocytes are very important in maintaining this blood-brain barrier or forming it. Okay, that's one of their jobs. They're also critical for providing regulation of ions and nutrients and gases. They will secrete, they will absorb neurotransmitters and will secrete chemicals, growth factors, and so forth to help keep the neurons healthy. And in fact, if you're ever in a lab where you're required to grow neurons in a petri dish, the neurons grow much, much, much better if they are grown on top of a layer of astrocytes. They live longer, they grow better, they form better axons and dendrites, and they're just much happier cells. Oligodendrocytes are rather funny. These are, oligo means many, and these are many branched. You can see one branch here, and you can see what it does is it's going to wrap its plasma membrane around multiple cells, more than one cell at a time. And that plasma membrane wraps around and around and around and around and forms myelin. A myelin sheath is designed to speed up the nerve impulses. We're going to spend significant time talking about the myelin sheath, so right now I'm just going to introduce this to you. And those oligodendrocytes are found in the central nervous system. Importantly, oligodendrocytes are not found in the peripheral nervous system. So keep that in mind. That sometimes shows up as a true-false question or something along those lines. In the central nervous system, axons that are coated in a myelin sheath look kind of white or yellowish because of the lipid content of the myelin. And so we call this myelinated structure, these myelinated axons, the white matter of the cell. Unmyelinated cell bodies of these neurons are our gray matter. Okay. Now I want to have you look at this section right here. Okay, you can see this little tiny section there. And there's another one right here. Okay. And you can see a little bit of a part right there. What those are is unmyelinated segments of axons. And those unmyelinated segments of axons are called the nodes of Ranvier. I'm sorry, Ranvier is the correct pronunciation. I always say Ranvier because that helps me spell it, but it's nodes of Ranvier. And these little areas of unwrapped axon regenerate the electrical signal that is passing down the axon. We will look at the details of that in a little bit here. For right now, I want you to kind of see those in this picture that you're looking at. The space in between here to here is called our internodal space. Okay? And you can see that that internode is covered in the myelin sheath. And so we go from node of Ranvier through this layer of myelin to the next node of Ranvier. That's what the electrical signal does. It's refreshed or regenerated in the nodes, and then it travels through the next segment of myelin. Gray matter, as I've mentioned already, are these unmyelinated cell bodies shown here. Okay, and so as we talk about the central nervous system, when you read about gray matter, when we talk about gray matter, that is what we're talking about, is these unmyelinated gray cells.